So let's see, we want to clone a gene bbk56 from plasmid pTDL11 into a cloning and expression vector pGEX3X. Let's open pGEX3X, which is in GenBank format, and plasmid pTDL11 again in GenBank format. And so I have opened the text panes of both these plasmids. And now I'll simply click on the graphic map to create graphic map of each of the plasmids. This is pGEX3X, and I'll go down and click on the graphic map of pTDL11 plasmid. So in dirty brown color we have our gene here bbk56, which sort of spans between 156th to 806th base pair within plasmid pTDL11. Now this is pGEX3X plasmid, which is a very common cloning and expression vector. And I'll just remove all the unique sites, so you can see the entire expression cassette is driven by track promoter, and at the end of the expression cassette we have a multiple cloning site where we can put our gene of interest for expression, and just before that we have a sequence which encodes for factor Xa recognition. So when the fusion protein is created, we can simply cleave it by factor Xa, right? And these are the enzymes 934 to 949. All these enzymes are single cutters which consist the multiple cloning site. So at the start we have BAMH1, at the end we have EcoR1, and let's imagine that we chose these two enzymes using which we are going to clone bbk56. So between BAMH1 and EcoR1. So the strategy is going to be very simple. We'll simply PCR amplify bbk56 gene with flanking regions containing BAMH1 and EcoR1, which we will subsequently cleave and use to clone in pGEX3X. But for that, the most important part is whether bbk56 has an inherent BAMH1 or EcoR1 site or not, right? So to check that, we'll go to particular sites and then we'll select for BAMH1 and EcoR1, and we can see here that bbk56 doesn't have any inherent BAMH1 or EcoR1 site. That means we have a green signal to use our strategy. So first we'll do PCR. Go at the top panel, click on PCR icon, and this is primer 1, details of primer 2. Here we select the DNA which we want to amplify. In our case it's plasmid pTDL11, so let's feed that. Now this is the forward primer, and this is oligo tail not identical to the target sequence part of primer, where this part is identical to the target sequence. So let's say this is forward primer and this is reverse primer. This is the sequence of forward primer which is identical to the target sequence. Remember, we are going to clone, we are designing primers for cloning, and just before that we are going to add GGA-TCC in case of forward primer, which is nothing but recognition sequence for BAMH1. In reverse primer we are going to add GAA-TTC, which is recognition sequence for EcoR1. And just so that our restriction enzyme doesn't falls off, we are going to add 4 more nucleotides GAGA. Similarly, we are going to do for the reverse primer. So this is the sequence which is not identical to the target sequence, that is bbk56 gene, whereas this sequence is identical to the target sequence, that is bbk56 gene, right? So now time to fill in the blanks. So I'll just copy BAMH1 site and GAGA, which is oligo tail not identical to the target sequence, and the entire sequence of primer which is identical to the target sequence in the box adjacent. Similarly, primer number 2 or reverse primer. Here we go. And then we have the sequence which is identical to the target sequence, which will go here. So we have pretty much filled up all the details for our PCR, and now we can just at the bottom right corner we have run PCR. We're going to hit that, and here we go. This is the PCR amplicon, PCR pTDL11 GenBank format, and we're going to create the graphic map, and let's try and confirm that we have added. So this is the bbk56 gene in the entire amplicon, and now I'll remove the unique sites, and from particular sites I'm going to choose BAMH1 and EcoR1 to confirm that flanking regions they do contain BAMH1 and EcoR1. So I'm going to select those, and here we can see BAMH1 site at the start of the gene, EcoR1 site at the end of the gene. So we'll save the amplicon as PCR pTDL11, and I'll just close all the windows and we'll start the construction. So go at the top panel and click on construct logo, and then select DNA1, which is always going to be vector fragment. In our case it's going to be pGEX3X. So we load this plasmid, and I'll remove all the unique sites and select particular sites, and from that we are going to pick BAMH1 and EcoR1. So this is BAMH1 EcoR1. Since the cuts are going to be in the clockwise direction, we'll first select EcoR1, then press shift, then select BAMH1. So here we go, we have EcoR1 on the left side, BAMH1 on the right side. The second fragment is PCR amplicon of plasmid pTDL11, which amplifies bbk56 gene. So again, from the particular sites we are going to select BAMH1 and EcoR1. Here we go. So on the left side we'll click on BAMH1, then press shift, click on EcoR1, and we have cleaved the amplicon. Now we'll just hit ligate. So this is our new construct. Let's create the graphic map. It is essentially pGEX3X plasmid containing BKK56 gene from 940 to 1590 base pair, and this is the entire insert from PCR amplicon of TDL11. So I'll minimize the text pane of new construct, and let's compare it with its parent plasmid, which is pGEX3X. So let's open pGEX3X, create a graphic map. I'll just take a little while to arrange both the graphic maps so that we can compare them well. So I'll remove the unique sites, and I'll just make BAMH1 and EcoR1 visible in both plasmids. So in case of pGEX3X, you can see that they both appear within the multiple cloning site, and in case of our new construct, we'll again choose BAMH1 and EcoR1 from particular sites. And we see that they flank the insert from PCR amplicon. So you can see the molecular weight of pGEX3X and that of the new construct, which has increased, and because of the insert from PCR amplicon, which is between 934 BAM-H1 to EcoR1. So once you confirm that you got the right construct, then you can save it. So open your text pane and then just hit save. Let's name the new construct as bbk56 gene in pGEX3X and save it. Now if you go to virtual cut, you can digest your new construct BK56 gene in pGEX3X with BAM-H1 and EcoR1. So we can see two bands upon BAM-H1 and EcoR1 cut. The larger band is 4964 base pairs. This is the one. And the shorter band is 657 base pairs, which contains bbk56 gene.